Okay, guys, so this lab's going to be looking at some of the common microbiological stains, some of the common different types of techniques and stains that will be used in a lab, some of the way how those stains work, and some of the main different types of categories of stains that you'll find in a microbiology lab. So the first thing that we need to talk about with viewing bacteria underneath a microscope is why we need to stain them and the two large types of broad different types of stains. So first things first, bacteria, if you put them underneath a microscope, you won't be able to see them. They are naturally colorless. They might have a little bit of a yellow, kind of a little slightly brown color to them, but when you zoom in on them on the microscope and you get them underneath the oil immersion, they will be completely colorless. Um, so stains allow you to see the bacteria. Stains are going to be some sort of dye, some sort of chemical, which are going to give the bacteria or the background some sort of color. A very common way that stains work is some sort of dye is added to the slide of bacteria, and it penetrates down through the cell wall. You can see the cell wall of the bacteria up here. This is the peptidoglycan. This is gram-positive peptidoglycan layer, and then their little cell membrane here. And you can see the bacteria here in the bottom, where this stain will penetrate down through the cell wall and either bond in the cell membrane or stick to the peptidoglycan inside of the cell wall, or bond to the DNA inside of the cell. Different types of stains work different ways, but that's some common ways that they work. But before we get too far into the different types of stains, we need to talk about the two different broad types of stains: a positive type stain and a negative type stain. This is a positive type stain right here, and this is a negative type stain right here. So positive type stain, what that means is you can see the individual bacteria here. This is one bacteria. This is one bacteria, one bacteria, one bacteria. You guys get the idea. The individual bacteria themselves, the stain has penetrated down through their cell membrane, down through their cell wall, and given the bacteria color from the inside of their cells. Whatever stain that's been used to give these guys colors has then been washed away, and the background of the stain remains clear, and the background or the bacteria themselves will remain colored. So you can see what color they are, and you can see what shape they are, and things like that. That's a positive type stain, color from the inside of the cell. Now, a negative type stain, on the other hand, is the exact opposite of this. This type of stain, you can see here, the bacteria are colorless. They have no color in them whatsoever, these little coccus-shaped bacteria here. The background has color to it. So what happens is you usually will put a little drop of things like India ink or some sort of nigrosin, some sort of really dark chemical, dark liquid on one part of the slide, and you put a little loop full of bacteria in that color and that stain, and then you just smear it across the slide. Now what this does is it colors the background of the slide. So the slide itself has black stain all over it. The black liquid, the bacteria do not uptake that black liquid. It doesn't penetrate through their cell walls. It doesn't penetrate through their cell membrane. So they just stand out on the background against that black background. So the light from the microscope passes through them since they're clear, but it doesn't pass through the background. Now over here you see the other thing. The light passes through the background, but not through the bacteria itself. So it's the other way, it's the opposite. For a positive type stain right here, and a negative type stain works the opposite. Positive, the color comes from the inside, and negative, the color comes from the background. So that's what the difference here. Positive type stain here, and a negative type stain here. So all the stains that we're going to talk about for this lecture are going to be positive type stains. So the very first thing that we need to talk about with staining bacteria is something called heat fixing. So bacteria, when you get them from a sample from your patient or a petri dish, they're going to be living. You're going to take a sample from your petri dish and you're going to put it on a loop. You can see that down here. They've got the culture here, a little petri dish or a culture broth tube or anything like that. You can see the petri dish up here. And they take a little sample from that and they spread it onto their slide or whatever. Now, that little sample is going to have living bacteria inside of it. As microbiologists, we don't want that. Living bacteria is not a fun thing to work with, especially underneath a microscope. You don't want to get living bacteria on the microscope. to cross-contaminate it. You also don't want to have the risk of dropping the slide and getting bacteria on the table or maybe on yourself or something like that. So heat fixing is going to do two things. You're going to put a little sample, little liquid. You may have to put a drop of water and put some bacteria in that drop of water if it comes from a petri dish. If it comes from a broth culture, you can just use the liquid itself from the broth. So heat fixing is going to do one of two things. It's going to kill the bacteria. We don't want living bacteria on our slide. That would be bad. So we're going to kill them. What we're going to do is you're going to add some liquid to that slide, and then you're going to run it over an open flame, either an incinerator to dry it or a Bunsen burner, anything at all that's put out a lot of heat. You can sometimes air dry them. You can put it on a slide warmer, lots of different ways to do this, but all you're doing is drying out the slide. You're evaporating all the water off. And what you're doing is you're killing the bacteria. This heat from the Bunsen burner, or the heat from whatever you're using, is going to evaporate all the water off, and it's going to kill the bacteria at the same time. It's too hot. The water is used to spread them around on the slide so they don't just make a big blob in the middle. You get a scoop of bacteria off your plate, you put it in the middle of a slide, and it's just going to be a good big blob. So you don't want to do that. So you put it in a little water droplet and spread it around and smear it around a little bit. And that's what they're called, smears, by the way. So anyway, the Bunsen burner is going to dry the slide, dry the liquid. It's also going to kill the bacteria. That's one thing that a heat fixing does. It allows the bacteria to die. The other thing that it's going to do is it's going to permanently seal them to the slide. Now, bacteria have a nice little sugary coating on their outside. And when you heat that sugary coating up, it gets sticky, it gets a little melty, and it's going to melt and ooze inside of the little tiny microscopic cracks that we can't see on this glass slide. This glass slide is not totally smooth, so these little sugary coating of the bacteria in there are going to get heated up. The sugar is going to ooze out and stick to the bacteria, or the slide, stick the bacteria to the slide. That's two things that heat fixing does. It kills the bacteria, and it's going to permanently stick them to the slide. Well, why do we want to stick them to the slide? We know why we want them dead. It's bad to have a living bacteria floating around. But why do we want them stuck to the slide? Well, if you didn't stick them to the slide, you wouldn't be able to put any stain on them. You'd put liquid stain on them or water to wash that stain off, different combinations of stain or whatever. And if they weren't permanently stuck to the slide, you'd just wash them away, which would kind of null and void the whole point of trying to stain them. You'd wash them away. So heat fixing is going to kill them, which is the whole point of the whole thing, so they don't get you sick or get anyone else sick or contaminate anything. And it's going to glue them to the slide. So once you've heat fixed every single slide, pretty much every single type of stain that's going to be considered a fixed mount, if you'll is going to begin with a heat fixing process of some form, a heat fixing process. There are other fixative processes that involve, instead of heat, they use acid, methanol, different types of chemicals that will permanently seal the bacteria to the slide. So there's other ways to do this, but heat fixing is the most common. So once you've heat fixed the bacteria to the slide, you've killed them, you've permanently stuck them to the slide, you can start adding liquid media, or liquid stains, I should say, excuse me, to that smear. And now they won't wash off. So the first type of stain that you will encounter in a microbiology lab is something called a simple stain. Now, a simple stain, as you can see here, is a positive type stain. It gives bacteria from the color, their color from the inside. Now, simple stains commonly use methylene blue, very common type of stain. This is what this blue color comes from. Another color here, you can see another methylene blue. This is probably crystal violet, actually. Methylene blue here, crystal violet, and safranin. Simple stains get their name because it just uses one stain, one dye, one liquid, one chemical. It is simple. It does not allow you to tell apart the gram reactions of the bacteria based on how their cell walls are put together. It does not allow you to see if they produce endospores. It doesn't allow you to see anything at all about the cellular makeup of this bacteria other than three things: how big they are, what shape they are, and what arrangement they're in. Are these gram-positive coccuses? I assume so. I don't know. Are they gram-negative? I also don't know. How do I know that I don't know? Because it's a simple stain. Everything that I stained is going to come out the same color. If they're gram positive or gram negative, it does not matter. So I don't know if these are gram positive or gram negative. I just know that they're caucuses. So it tells me their shape. It also will tell me their size. How big are they? Well, you have to look at quite a few bacteria underneath the microscope to be able to judge apart size. You'll see here these are rod-shaped bacteria that have all been simple stained with different simple stains. They're all the same size, but if you look up here, these are also rod-shaped bacteria, and these are smaller than those. So these are also quite small. You just have to look at quite a few bacteria underneath the microscope before you can successfully judge size. So you get shape. You'll be able to see the shapes of bacteria here. There are three common shapes. You have coccus, round, rod or bacillus, which is a tic-tac sort of shape, kind of like a hot dog. You see that here. These are our coccuses, the little round balls. And then spirals, which kind of look like a spiral pasta noodle or a little twisty thing underneath the microscope. So what you'll be able to see is what shape they are. Are they round? Are they rod-shaped? Or are they spiral? You'll be able to tell how big they are. Are they little like this guy? Or are they large like him? You'll be able to tell that. And then you'll be able to tell one more thing is the arrangement. Do they come in clusters like this? This is Staphylococci arrangement, not the species, the arrangement. This is chains of bacillus, streptobacillus, little lines of bacillus, same up here. You can have random assortment where they're just blobbed across the table, blobbed across the slide. You can have tetrad arrangement, lots of different arrangements. So the way that the bacteria are arranged is going to be very helpful for diagnostic purposes as well. So you get size, you get shape, and you get arrangement with a simple stain. The only problem here is you do not get gram reaction, which is the whole point of this. This is a useless stain for diagnostic purposes. This is a quick down and dirty, I just want to see what it looks like underneath a microscope. You do not want to diagnose anyone with a simple stain. This is just to see what it looks like underneath a microscope. Also, a lot of beginner courses start with this for microbiology just because it's pretty easy to do. There's not a lot of complicated steps inside of this. A lot of simple stains like methylene blue are going to bond inside of the cell to DNA, cellular DNA. Crystal violet will bond to peptidoglycan. It just kind of depends on the dye that's used, on this one stain that you select. That's why it's called a simple stain, because it is one single liquid. And all you get from this are size, shape, and arrangement. You don't get gram reaction, which is the most important part of staining because that allows you to continue diagnosing what type of infection or bacteria your patient has.